For over 2,000 years, they sat hidden in the desert, rolled up, sealed in clay jars, buried in caves near the shore of the Dead Sea. And when they were finally discovered in 1947, they became one of the most important archaeological finds in human history. The Dead Sea Scrolls, the oldest known copies of the Hebrew Bible, ancient calendars, community rules from a mysterious religious sect, prophecies, psalms, forgotten texts that never made it into any modern Bible at all. But here's the problem scholars have wrestled with for 70 years. The Dead Sea Scrolls aren't really scrolls, not anymore. What archaeologists actually pulled out of those caves was roughly 25,000 tattered fragments, a shredded, scattered 2,000-year-old jigsaw puzzle with no box, no picture, and no guarantee that every piece even belongs to the same puzzle. For decades, scholars pieced it together the only way they could, by studying handwriting styles, ink patterns, and the shape of torn parchment edges. Educated guesswork, dressed up as science. Then, in 2020, a team of researchers did something no one had tried before. They didn't study the ink. They didn't study the handwriting. They studied the skin the words were written on. Because the Dead Sea Scrolls aren't paper, they're leather. Ancient animal hide. Which means, hidden inside that parchment, preserved for 2 millennia, was something scholars had never thought to look for. DNA. Actual genetic material, sitting quietly inside 2,000-year-old goat and sheepskin, waiting for someone to finally ask the right question. And when they did, what they found didn't just help rebuild the puzzle. It rewrote what we thought we knew about who wrote these texts, where they came from, and how the Bible itself came to exist in the form we read today. This is the story of how scientists put the Dead Sea Scrolls under a microscope built for genetics, and why the results shocked even the experts who ran the study. Think about what that actually means for a second. For 70 years, some of the smartest biblical scholars on the planet had been trying to solve this puzzle using their eyes, their training, and their best judgment. Handwriting analysis, ink chemistry, the color and grain of ancient leather. All useful, all limited, all, ultimately, guesswork dressed up in academic language. Genetics doesn't guess. A DNA fingerprint either matches or it doesn't. And when scientists finally ran that test on fragments that scholars had confidently grouped together for decades, some of those groupings didn't survive contact with the evidence. Pieces that looked like they belonged to the same scroll turned out to come from entirely different animals, which meant entirely different scrolls, written by entirely different scribes, possibly in entirely different parts of ancient Judea. That's the shocking part. Not that scientists found DNA in old leather, that the DNA proved some of our long-standing assumptions about these texts were simply wrong. To understand why this mattered, you need to understand exactly how broken this puzzle really was. The scrolls were discovered between 1947 and 1956, mostly in 11 caves near an ancient site called Qumran, on the northwestern edge of the Dead Sea. Bedouin shepherds found the F.S.A.R.A., earthbatch by accident. Archaeologists found the rest over the following years. In total, roughly 25,000 fragments emerged from those caves, some as large as a full scroll, many no bigger than a postage stamp. Scholars have spent more than 7 decades trying to sort these fragments into coherent manuscripts. Which pieces belong to the same scroll? Which fragments are from different copies of the same book, written by different scribes in different places at different times? For most of that history, researchers had only one method available: paleography, the study of handwriting style, combined with physical clues like the color and thickness of the parchment. The problem is that paleography is subjective. 2 scholars can look at the same handwriting and disagree, and torn, damaged, sun-bleached parchment doesn't always give you a clean signature to compare. Some fragments simply couldn't be confidently placed anywhere. Worse, some fragments that looked like they belonged together, because the handwriting matched, or the parchment looked similar, may not have actually come from the same scroll at all. This is where the animal skin becomes the real story. The scrolls weren't written on paper. They were written on parchment made from the tanned hides of sheep, goats, and occasionally cows, which means every single scroll fragment is, biologically speaking, a piece of a once living animal. And living animals carry DNA in their skin cells, DNA that, under the right conditions, can survive for thousands of years. In 2020, an interdisciplinary team from Tel Aviv University, the Israel Antiquities Authority, and Uppsala University in Sweden decided to test that theory. The project was led by Oded Rechavi, a molecular biologist who studies genetics in microscopic worms, and Noam Mizrahi, a scholar of ancient Hebrew literature. The two met by chance, while sitting next to each other on a bus during a university orientation event. 8 years later, that chance meeting turned into one of the most ambitious scientific studies ever performed on the scrolls. Their goal was simple to state and brutally difficult to execute: extract genetic material from the scroll fragments without damaging the priceless text written on them, identify which animal species each piece of parchment came from, and use that genetic fingerprint to figure out which fragments were biologically related and which ones only looked related on the surface. To do this without harming the manuscripts, researchers didn't touch the inscribed surface at all. Instead, they carefully collected microscopic scroll dust, tiny particles that had already flaked off the uninscribed back of the fragments over the centuries. That dust was sent to a specialized ancient DNA laboratory in Uppsala, Sweden, one of the few facilities in the world equipped to handle genetic material this old and this fragile. Researchers worked in full body, protective suits to avoid contaminating the ancient samples with modern DNA. Their own skin cells, hair, or breath could easily ruin thousands of years of preserved genetic evidence. The project took 7 years from start to finish, and when the results came back in 2020, published in the scientific journal Cell, they didn't just help reassemble a few torn fragments. They challenged assumptions scholars had held for generations about where the scrolls came from and who was really writing them. Consider the sheer scale of what these researchers were attempting. Ancient DNA is notoriously fragile. It degrades over time, it fragments into tiny, unreadable pieces, and it's incredibly easy to contaminate with modern genetic material. A single stray skin cell from a researcher's hand, a cough near an open sample, even bacteria from the surrounding soil could flood the sample with modern DNA and make the ancient signal impossible to isolate. That's why the extraction work had to happen inside a specialized paleogenomics lab in Sweden, under conditions closer to a high-security virology unit than a typical archive. And this wasn't the only scientific angle running in parallel. While Rachavi's team in Uppsala focused on the animal DNA embedded in the parchment itself, a separate team, led by a genomics researcher in New York, was simultaneously studying the microbial residue on the scrolls, the bacteria, fungi, and other microscopic organisms that had colonized the parchment surface over 2 millennia. Together, these two lines of research turned the scrolls into something scholars had never had before, a genetic archive, not just a textual one. The stakes of getting this wrong were enormous. These are among the only surviving primary sources for understanding Judaism and early biblical text in the centuries surrounding the birth of Christianity. Misidentifying which fragments belong together doesn't just create an academic inconvenience, it can distort our entire understanding of how sacred texts evolved, which communities produced which versions, and how a scattered set of ancient writings eventually became the foundation of religious traditions followed by billions of people today. The first finding was almost mundane on the surface, but it set the foundation for everything else. Researchers successfully extracted usable ancient DNA from 26 separate scroll fragmi NTs. Of those, 24 came from sheepskin, 2 came from cowhide. That might sound like a minor detail until you realize what it allowed scholars to do next. Take the book of Jeremiah. For years, certain fragments believed to be part of the same original scroll were treated as a single, continuous manuscript. But when researchers ran the genetic tests, they discovered that some of those matching fragments weren't even made from the same species of animal. One piece was sheepskin. Another, believed to belong to the exact same scroll, was cowhide. That is not a subtle difference. It means these fragments could not have come from the same original document. They were from entirely separate copies of the book of Jeremiah, meaning multiple divergent versions of this biblical text were circulating in ancient Judea at the same time. This might be the single most important discovery in the entire study. It suggests that the Hebrew Bible as we know it today was not copied from one single, stable, agreed-upon master text. Instead, different communities were eating, copying, and preserving different versions of the same sacred books, sometimes with real textual differences between them. The Bible during this period wasn't one fixed book. It was a living, evolving collection of texts, still being shaped and reshaped by different scribes in different places. The second major discovery involved geography. Prior to this study, many scholars assumed the Qumran community, often linked to a Jewish sect called the Essenes, produced most, if not all, of the scrolls found in the Qumran caves themselves, locally in one relatively isolated location. The genetic data told a different story. By comparing the DNA signatures of different fragments, researchers found that some scrolls at Qumran were made from animals that likely never lived anywhere near Qumran. These scrolls, in other words, were not written locally. They were brought in from elsewhere in ancient Judea and added to the collection. This matters because it changes how historians understand the Qumran community itself. If the scrolls at Qumran represent texts gathered from a much wider geographic and cultural network, not just the output of one isolated desert sect, then the Dead Sea Scrolls collection is a far more representative snapshot of Jewish religious life across the entire region during the Second Temple period, rather than the narrow output of one insular group. A third finding involved a scroll fragment containing text from the book of Isaiah. This fragment had long been cataloged and published as a Qumran find, but its genetic signature didn't match the other Qumran scrolls at all. Its DNA fingerprint pointed somewhere else entirely, somewhere researchers haven't been able to definitively identify. As lead researcher Noam Mizrahi put it, the finding raised an uncomfortable new question. Was this fragment actually found in the Qumran caves at all? Or did it originate somewhere else, a location that remains unidentified to this day? Finally, researchers used the same genetic method on a well-known liturgical text called the Song of the Sabbath Sacrifice, of which multiple copies were found both at Qumran and at Masada, the famous mountaintop fortress where Jewish rebels made their last stand against Rome. Historians had long assumed the Masada copy was simply brought there from Q. Moran, since the same text appeared at both locations. The DNA said otherwise. The Masada copy's genetic signature didn't match the Qumran copies. It came from a different animal, likely produced somewhere else entirely, meaning this text was independently circulating across ancient Judea rather than spreading outward from a single source at Qumran. Each of these findings sounds narrow and technical on its own. A fragment here, a mismatched hide there. But stacked together, they add up to something much bigger. Solid, biological, physical proof that the story of how the Bible came together was messier, more decentralized, and more geographically widespread than scholars had assumed for the better part of a century. There's another layer to this that's easy to overlook. Before this study, when scholars found two fragments with similar handwriting and similar-looking parchment, the default assumption was often that they belonged to the same original document unless there was strong evidence otherwise. The genetic testing flipped that. Logic. Now, similarity in handwriting alone isn't enough. A fragment has to pass a biological test before it can be confidently grouped with another. That's a fundamentally stricter, more rigorous standard than anything scholars had access to before. And it means some long-accepted groupings of fragments will likely need to be re-examined using the same genetic approach in the years ahead. It's also worth remembering just how small these samples were. Researchers weren't cutting chunks out of priceless ancient manuscripts. In man-eye cases, they were working with literal dust, microscopic particles that had already broken away from the unused backside of a fragment over the centuries. XT, rating a usable, reliable genetic fingerprint from something that's small, that degraded, and that old, is itself a remarkable technical achievement, independent of what the results actually revealed. For decades, one of the biggest open questions in biblical scholarship was this: how uniform, or how diverse, was Jewish religious literature in the centuries just before and after the birth of Christianity? And was there one dominant, standardized version of Scripture, quietly enforced by religious authorities? Or were there many competing versions of these texts, circulating simultaneously among different communities, each slightly different from the next? The Dead Sea Scrolls were already part of this conversation, before DNA testing ever entered the picture. Textual differences between certain scroll fragments and later, more standardized versions of the Hebrew Bible had already hinted that ancient scribes weren't working from one single, tightly controlled master copy, but without a reliable way. To prove which fragments were truly independent documents versus damaged pieces of the same scroll, that argument remained partly speculative. The genetic evidence changes that. It provides hard, physical proof, not just literary analysis, but biological fact, that different, independently produced copies of the same biblical books, containing real textual variations, were circulating across ancient Judea at the Sus, AIM time. This is a serious problem for any assumption that there was ever one clean, agreed-upon, unchanging version of these texts in this period. Instead, what emerges is a picture of active, ongoing scribal culture. Multiple communities, in multiple locations, each producing and preserving their own versions of sacred literature, sometimes years or decades apart from each other, all before anything like a fixed canonical Bible existed. It also reshapes how historians view the Qumran community itself. If the scrolls found there weren't all produced locally, but instead represent texts gathered in from a broader network across the region, then Qumran functioned less like an isolated monastery producing its own private religious library, and more like a kind of archive, a collection. Point where texts from many different Jewish communities across Judea ended up being stored together. That's a meaningful shift in how scholars think about who these people were and what role they played in the wider religious landscape of their time. Beyond the historical and religious implications, this study also proves something enormously important on a purely scientific level. Ancient DNA can survive in processed animal skin parchment for 2,000 years, and it can be extracted and analyzed without destroying the priceless historical text written on top of it. That is a breakthrough with implications far beyond the Dead Sea Scrolls. Historians and archaeologists have thousands of other ancient parchment documents around the world. manuscripts, legal documents, historical records, where the same DNA-based technique could potentially be applied to solve completely different historical puzzles. Researchers involved in the project have been careful to note that this study didn't answer every question about the scrolls. If anything, it opened new ones, like the mystery of that Isaiah fragment with no clear genetic match to anything else at Qumran. But that's exactly how meaningful scientific research is supposed to work. It doesn't hand you a tidy, complete answer. It gives you sharper tools, points you toward the next unresolved mystery, and forces you to keep digging. It's also worth being careful about how far to take these conclusions. Some commentators have overstated what the DNA evidence actually proves, treating it as definitive confirmation that entire finished scrolls, rather than raw, unprocessed parchment, traveled long distances across ancient Judea. That's a bigger claim than the genetic data alone can fully support. The DNA tells us where an animal's hide likely originated. It doesn't, by itself, tell us with Siri. Tainty where the parchment was tanned, where it was inscribed, or how many hands it passed through before ending up in a cave near Qumran. Responsible scholars treat the genetic findings as one powerful piece of evidence among several, alongside paleography, radiocarbon dating, and textual analysis, rather than a single silver bullet that settles every debate on its own. That combination is really the point. No single method, not handwriting analysis, not radiocarbon dating, not genetic testing, can fully reconstruct a 2,000-year-old library on its own. But layering these methods together gives historians something far more powerful than any one technique alone, a way to cross-check assumptions, catch errors that any single method might miss, and build a more reliable picture of how these texts were written, copied, and passed down across a scattered and diverse ancient world. What started as a chance conversation between a worm biologist and a Bible scholar sitting next to each other on a university shuttle bus turned into a 7-year research project that used cutting-edge genetic science to answer questions historians had been arguing about since the scrolls were first discovered in 1947. The results didn't just help reassemble a handful of torn fragments into their correct manuscripts. They proved, on a biological level, that ancient Judea was home to multiple independently produced versions of the same sacred texts, circulating across a much wider geographic area than scholars had assumed. They showed that Qumran, far from being an isolated source of scripture, was more likely a gathering point for texts pulled in from across the region. And they left behind at least one unsolved mystery, a scroll fragment whose genetic signature doesn't match anything else found at the site where it was supposedly discovered. The Dead Sea Scrolls have survived wars, empires, earthquakes, and 2,000 years buried in desert caves. And in 2020, for the first time, science reached past the ink and the parchment and read something the scribes themselves never knew was there. A genetic signature hidden in the skin, quietly recording the truth the whole time. If you found this breakdown interesting, that's exactly why this story keeps getting told. Because sometimes the biggest historical breakthroughs don't come from finding something new. They come from finally knowing how to ask an old artifact the right question. And this is very likely just the beginning. The technique developed by this team can, in theory, be applied to any of the thousands of still unidentified Dead Sea Scroll fragments sitting in archives, waiting for someone to figure out where they belong. Every one of those fragments carries the same kind of hidden genetic fingerprint. As the technology gets faster and cheaper, it's entirely possible that more of that 25,000-piece puzzle gets solved, not through a lucky guess about handwriting, but through the quiet, undeniable evidence written into the skin itself, 2,000 years before anyone thought to look for it.